Every year in Africa, 3 million cattle succumb to African animal trypanosomosis, a debilitating disease of livestock that is caused by trypanosome parasites. These parasites are mainly transmitted by tsetseflies as well as by other biting flies. Known as nagana in cattle, goat, and sheep, and as sura in camel, the disease's main symptoms include anemia, fever, swelling of the lymph nodes, and decreased appetite. It leads to weight loss, lethargy, abortions, a suppressed immune system, and increased chances of concurrent infections. The productivity of infected animals is reduced, and thus the availability of livestock products and services like tilling of agricultural land. Production costs are escalated due to investments for treatment. African animal trypanosomosis causes direct and indirect losses of approximately $4.75 billion per year in Africa. Researchers from ICIPE have developed a much-needed, non-invasive, rapid, affordable, efficient, and easy method to diagnose African animal trypanosomosis. As a result, through a simple urine test, which can be administered even by livestock keepers, it is possible to confirm whether an animal has the disease. Dr. Merit Getahun, ICIPE scientist and leader of the study, explains the process to the discovery, starting with an overview of the challenges in diagnostic strategies.
We can treat the livestock. We have trypanosome drugs there. We can also do vector control. You can mass trap the vectors, so that you can avoid. We also have repellent, which is developed by ICIPE, so that you can minimize the contact between the livestock and vector. But the other one is, you can't protect or repel or trap each and every fly. You also need to treat the livestock, which is based on diagnostics. We do have two techniques. We have microscopy, which we will use it in the field, so that you need to take blood. There are some steps that we have to follow. It's a nice technique, but it's not so sensitive. Most of the time, you need to have a high parasite load to see it. The other one is we have PCR. PCR is also too technical again. And of course, both of them have the expensive, because these are all detect an active infection. But if you want to detect an antibody, we have another technique, but we need to have a technique that detects active one. We work with pastoralists and farmers, and you see how they struggle to keep their livestock health. We don't have simple diagnostic. So then I started to challenge myself. Is it possible to simplify this diagnostic, so that these pastoralists can make decisions based on knowledge? At the moment, they just do it based on symptoms. Symptoms are not specific, so that this will result in livestock mortality as well as selective pressure for resistance development. The hypothesis as a chemical ecologist is infection, or like say this animal trypanosomiasis, modifies host odor, like livestock odors will change due to infection. When a parasite transmitted by vector and goes into blood of a cow, there is a fight between the parasite and the host. The parasite tries to establish itself in the cow, by suppressing immune system, but the cow also fight back. We studied the odor of healthy animal, let's say a cow, and then we profiled all of those odors, and we also did the same for a sick cow, example from trypanosome, and we look for any new odors that are come due to the sickness, or any that has changed in the concentration. Odors can be measured in various ways. Then we found urine is as the simplest, non-invasive, but at the same time it also represents quite fairly represent what is going on inside the animal body, so that it can tell us about the health status of the animal. Once we get the urine from an animal, we will trap its odor, and the trapped odor will be injected into GCMS. This is done to separate the different compounds that are within the mix in the sample, and also to identify who they are. So what you see here is called a chromatograph. Different compounds present in cow urine sample. These are the results from two different cows, healthy and sick. We see in majority they have similar odor profile. But let us look at this peak, which is one of the phenolic compound. It is the same compound, but there is a pretty big difference between sick and healthy. That means there is more phenolic in the sick cow than healthy cow. Here is another example. Let us look at this peak, which is one of the ionon. We cannot see it in the healthy cow, but produced when a cow is sick. So result like this is exciting as it tells there is a difference, or we call it molecular phenotype, between healthy and sick cow. And it likely has something to do with the infection, and we can use this biomarker for disease diagnostic. We have identified the biomarkers, especially the phenolic. Now we want to use these phenolics to make disease diagnostic, so that we have to detect those biomarkers inside the livestock urine without, with the simple technique, which we can see by our eye. The first step is to get livestock urine. Then dilute 1 ml urine in 9 ml water. Mix well. Then add 1 ml urine solution in 5 ml Folin reagent, followed by addition of small amount of sodium carbonate to activate the reaction. Mix well and observe color change. If the urine sample obtained from trypanosome infected animal, the color will change to blue within a minute. If not, remains green. Often, the more the parasite load, the more intense the color change. We test first, we used the control experiment. Everything is controlled. We also compared our technique with microscopy, with the PCR, so that we showed that it's very comparable with the PCR. Like, for example, we got samples which are asymptomatic. You don't see with the microscopy, but with the PCR they were positive, and then we test the urine with our biomarker, they were also positive. That is shows that how sensitive it is. The technique also detects an active infection, because we have tested the treated animals afterwards and they were negative. So it really shows that it detects an active infection. We also tried to see how specific it is. So we have got some animals, like about 5 animals, which were sick. They have a low PCV, like anemic, and by all means you classify them as a sick. But when we test the urine, they are negative. They are of course negative for TRIPS, where microscopy and PCR, as well as with our biomarker. But when we do the detailed analysis, they were sick from anaplasmosis and tellurosis. That means our biomarker-based diagnostic is selective or specific, but of course we need to challenge with more related diseases in the future to really tell it's really specific to animal trypanosomiasis or not. We have now the ingredient. We know which one, the reactors, the reagent we need. We also know the biomarkers. So it can easily be developed, with the right partner and also investors, that we can develop into a paper dipstick. We have for let's say for pregnancy test. We also have for diabetic test, similar things. So that if we impregnate this reagent inside those special paper, so that you just only dip this paper in the urine, and then you see the result. And in that way, I think it becomes more pastoralist or farmers friendly and accessible as well. We can also reach more pastoralists by doing that way.
This simple novel diagnostic tool for African animal trypanosomosis is emblematic of ICIPE's goal of developing science-led, inclusive innovations that are effective, accessible, and affordable to all segments of society. It will also support the goal of sustainable livestock farming in Africa.